In this section of M1.3.1, let us consider the ethical guidelines in animal research. We are all aware of the model animals such as rats, mice, zebrafish, Drosophila, to name a few, that are extensively used in the various scientific research fields due to their high genome similarity with the humans. There are also other higher animals such as primates, cattle, used in the animal research such as veterinary and biomedical research. The animal research has gained importance in various The animal research has gained importance in various research areas such as toxicology, drug discovery, understanding of the various diseases at the genetic and molecular level. Most of these are for the benefits of the humankind. However, animal research should not compromise with the welfare of these animals used for experiment purpose, because unlike human subjects, these are not able to give their consent for participation in such studies. The humanest possible treatment of experimental animals, far from being an obstacle, is actually a prerequisite for successful animal experiments. This has been mentioned by Russell and Birch, the two scholars from the university's Federation of Animal Welfare, in their publication Principles of Humane Experimental Technique in 1959. Herein, they laid out principles of three R's: replacement, reduction, and refinement. Replacement: the substitution of insentient material in place of conscious living higher animals used for experimentation. Reduction: reduction in the number of animals used to obtain information of given amount and precision. Refinement: any decrease in the severity of inhuman procedures applied to these animals which still have to be used. Russell and Birch described these three R's in this order, reflecting the order in which they should be addressed. Replacement: the sentient animals should not be used if non-sentient alternatives are available. Reduction: if animals do have to be used, then the project's design and analysis should be such that the minimum number of sentient animals are used compatible with achieving the objectives of the research. Refinement: appropriate measures should be taken to mitigate any pain, suffering, or distress that the animals might feel. The Guiding Principles for Medical Research Involving Animals, published in 2012 by the Council for International Organization of Medical Sciences and the International Council for Laboratory Animal Sciences, state the principles of the three R's: replacement, reduction, and refinement should be incorporated into the design and conduct of scientific and/or educational activities that involve animals. Thus, these three R's have become synonymous with the measures to improve the welfare of animals used in research and are now used as an ethical framework for improving laboratory animal welfare throughout the world. Various countries have regulations and laws for ethics pertaining to animal research based on their legal and cultural backgrounds. These are expected to be followed scrupulously by the research institutions and the researchers. The following aspects should be stringently addressed while undertaking the animal research. Procurement: the animals should be procured from the authorized breeding facilities which maintain healthy conditions of housing as well as ensure the proper nourishment of these. While shifting these to the research centers, they should be transported under hygienic conditions, taking care that the animals will not be subjected to stressful conditions. Animal housing and maintenance facility: the research center must have a certified animal housing facility which is duly recognized by the Animal Ethics Committee as per the norms of the country. Such national bodies will have their guidelines specified for various experimental animals and the type of research undertaken using them. Following of these will be ensured by the nominated members of the Animal Ethics Committee constituted for that particular research center. The facility must have separate areas for newly procured animals to be quarantined, their subsequent acclimation, the treatment period, and the experimentation technique. Optimum conditions. The animal housing facility should maintain daily optimum conditions such as light and dark cycle, ambient temperature, humidity, and other factors as per the natural surroundings of the respective animals housed. If it is an aquatic animal housing facility, such as fish breeding and maintenance facility, the water parameters like pH, ammonia, nitrates and nitrites, hardness, conductivity, dissolved oxygen should be checked daily. Proper records should be maintained daily by the specially trained staff. Disposal of dead animals. This should be done with correct norms pertaining to biosafety and preferably should be discarded along with the other biohazardous material through a proper authorized agency. Care taken during experimentation. All efforts should be made to make sure that minimum pain and suffering is caused to the animals while performing any techniques such as injections or drawing out of blood. They should be handled with utmost care and may be anesthetized as per the guidelines. Sacrificing the animals should be done in an ethical way as prescribed in the protocol specific for the types of experimental animals. OECD guidelines should be followed for various toxicity studies. Ethical clearance. The research studies involving animal experimentation should be approved by the respective Animal Ethics Committee, which will look into the aspects such as justification of animal usage, minimum number of animals to be used, consider if there are any other alternatives such as in vitro studies or in silico studies possible, etc. All in all, we, as researchers, must understand the ethos behind the animal experimentation, that it is used for the benefit of humans. Therefore, the responsibility lies on us, humans, to treat these animals in a judicious and, more important, in a humane manner.